I'll be continuing with what Scott talked about with collecting evidence and what Rhonda talked about with processing items in a lab to produce data. But all that collecting and processing is pretty useless or uninformative if you can't use the data to actually produce information. So what I'll be talking about is how do we automate the production of information from all of those samples and that data. So to do this and to make this a little more familiar than hitting you with acronyms in technology, mathematics, and chemistry and all kinds of science, I'm going to introduce a fictional character, Sarah. So meet Sarah. Sarah, we fictionalize or imagine, was in the World Trade Center when these events happened, and we're going to be tracing how we identify her using DNA technology. So during the World Trade Center attack, over 2,700 people went missing, and most of them had died. And from this, and I'm referring back to the time when I did the analysis in this case at Cybergenetics about 15 years ago, there are about 18,000 victim remains that had been collected. So that's what we're starting with. There's this attack, massive incineration of the people, the contents, and we have collected victim remains. Now the task is to identify those victim remains of Sarah and the other 2,700 missing people. But their remains are not like remains from a car crash. They're basically, they look like often just charred pebbles. There's not much there. So the task is to associate the victim remains, these charred chunks of remains, with the people who've been reported missing. And the reason that the medical examiner wants to do this is so they can confirm the death, but also to bring closure to families and return the victim remains to the family. So a big idea here is that we can use biological evidence, that the same DNA is in the victim remains as it is in the missing person. That is, you have Sarah, and she's a missing person, and there's DNA from her as a missing person. There's also DNA that she would have left in a victim remain. And so the approach is to compare the victim remains DNA with the missing person's DNA and connect them through the common DNA of the same person. Okay, that sounds pretty straightforward, but this is not your grandmother's DNA. It's not the way DNA was thought of 20 years ago when you have perfect DNA evidence like blood on a floor from one person and you have a suspect and you're trying to connect them. The DNA is highly compromised or non-existent even. From the victim remains, we don't have blood or DNA under a fingernail or a sexual assault kit. We have charred victim remains. There's not much there. The DNA is damaged, it's chopped up, it's hard to even get data from. And then the missing people don't make good DNA references because they're missing. There is no DNA from the missing person like you would have in a crime from a suspect or a victim. There's no DNA from that person that we can compare with. We do have their relatives, we do have personal effects, sweaters, toothbrushes, and so on. And so we need a much more sophisticated DNA approach to take the data that's being collected from DNA to associate the victim remains with the missing people. So the key idea here is the genotype. So I'll tell you what a genotype is. In nature, Sarah would have inherited 2 genomes, a set of chromosomes. One set of chromosomes from her mom and one from her dad. And in science, forensic scientists don't sequence all 3 billion letters of the genome yet, But instead, they test maybe a dozen different locations, short paragraphs of text on our DNA, a few hundred letters long, to find out the pair of inherited alleles. An allele is a variant, so at a particular genetic location or locus, there can be many different DNA sequences that can be there, like genes. Those variants are called alleles. And for regular autosomal DNA, which is most of our DNA, at each genetic location, we have a pair of alleles, one from our mother and one from our father. And that is the genotype at one locus. So Sarah, at each of these dozen or so tested genetic loci, these locations, has a pair of alleles, one from each parent. That's her genotype. And instead of drawing you lots of pictures of data or math or figures or probability distributions, I'm just going to use Sarah's picture. So this is a picture of Sarah. You can see here, it's a clear picture. It's not fuzzy. It's not grayed out. And let's just use her face for the purpose of this talk so you can see what I'm talking about as her genotype. So this is Sarah, and she's going to be represented through her genotype, which will be her picture for this talk. Well, when we look at DNA, whether it's from a cheek swab, from a reference sample, or for charred victim remains, or from any sort of DNA from a crime scene, there's only maybe 50 or 100 cells there. There's not much DNA. We can't see it. It's not detected. That DNA has to be amplified. And so what revolutionized forensic DNA testing in the 1990s was polymerase chain reaction, or PCR, the same way of amplifying DNA that you're familiar with from COVID testing. And PCR, in theory, keeps doubling the number of copies of DNA. I show here in blue if you have two alleles, an A and a B, whatever they are, but two different variants at a location. After one cycle, you have two A's and two B's. It's doubled. And then you run it for another cycle, and you have four A's plus four B's. You've doubled the alleles, the DNA copies. And then 8 A's and 8 B's, and 16 A's and 16 B's. You do that 28 times, and you get billions of copies. They're labeled, they're detectable, and if everything worked perfectly, the picture on the right, the heights of those purple bars are showing you how much you have. You've got a lot of DNA, it's detectable, and it's the same amount. And that's what goes on in the mind of many analysts. But that's not what actually happens in the test tube. PCR is an imperfect amplifier, like stereo amplifiers if you turn them all the way up. It's not faithful. It doesn't always copy. It may copy 70% of the time, not 100% of the time. And so this process of copying 28 times and failing with some probability, like 30%, makes PCR a random process. It's inherently random. That is the copying mechanism that lets us even see the DNA. And so in the bottom, what I show is if it's not always doubling, in red you may have A plus B is what you're starting with, allele A and allele B. And then maybe you get lucky, A and B both copy, you get two A's plus two B's in red. But now one of the A's doesn't copy, you get only three A's and four B's. And then some of the copying works, some doesn't. You have 5 A's and 7 B's. And you continue when it's all done. The amount of the allele that you observe, how much DNA, is related to what you started with, but has a lot of random variation. And that random variation stops most DNA analysis by human review and the older methods that were in use 20 years ago. Now, according to the science, not how people might try to solve the problem, the right way to infer genotypes from data is to say, well, anything's possible, but the data are telling us what the probabilities are. That's the science. But what was done, and is still done by most labs, is more of an absolute type of genotyping. And either it's obvious to the analyst or it's not, or it's obvious to a primitive computer program or it's not. And that type of old human absolute review needs a clear answer. And on this type of data, it's not going to happen, and that will fail. However, coincidentally, in 2001, I filed a patent, which later issued, and invented a way of working out what all those probabilities were by determining what the PCR copying randomness was for each experiment directly from the DNA data, and that way we could get accurate genotype probabilities. And that's been called probabilistic genotyping. As Rhonda said, most labs will be doing that soon, using some product or another. And what that does is it says it's not just one allele pair or no allele pairs, or I don't know, or it's inconclusive. It's every allele pair, every possibility of a genotype with some probability. And so what I'm showing here on the bottom left is Sarah's probabilistic genotype from the evidence. It's a little bit blurred, a little distorted. I'm showing the evidence that came from the particular charred victim remain. So that's going to be our picture that you can see of what her probabilistic genotype from her victim remain looks like as a picture. It's a little distorted, it's not exact, it comes from the victim remain. Now, the purpose of inferring a genotype is to be able to make a comparison. And if we had a reference genotype, which we don't, we could take an inferred genotype from crime scene evidence, like victim remain from the World Trade Center, or a sexual assault, or something that is evidence, and compare it with a reference genotype, and we can measure the strength of match. That's a match statistic. For the forensic cognoscenti here, that's called a likelihood ratio, or an LR. I won't be talking about that in any great detail. That's the match strength. And you see that we can compare an inferred evidence genotype from Sarah on the left from her victim remain, that little gray round thing, with her reference genotype if we had it. And what the match statistic is, and there's no math here, but what it is a certain chance of seeing her genotype in the population. I call that P. It's pretty standard. The population genotype, the prior, what you think you have as the chance of seeing her genotype randomly before, by coincidence. After the data is examined, you end up with a probability of that genotype. Remember, every genotype has a probability, so you take the ratio. After you see the data, that's Q, divided by before seeing the data. What's the change in probability from coincidence to what the data is telling you? That's the match statistic. If it's a big number, 6 zeros, like a 1 followed by 6 zeros or a million, that's showing a strong association. If it's a small number, like 1 in a million, that's 1 over 1 followed by 6 zeros, that's showing that it's probably not her. So in the end, what we have is one number, the match statistic. And that's the end game for forensics, for DNA, whether it's in court, whether it's for mass disasters. What is the strength of association using the standard match statistic, comparing what you know after you've analyzed the data compared with before? And that is what lets us make comparisons. And I'll spare you the details today. Okay. So when we're looking at the World Trade Center victim remains, we're really asking the question of these 18,000 charred pebbles, these charred pebbles, which one, or which several, came from Sarah? Is it the one on the left? Is it one on the top right? The one on the bottom right? Is it none of them? Which one came from Sarah? That's what we're trying to do, is to identify which one or two or none are her victim remains. Now, from these 18,000 victim remains, a lot of testing was done by different companies, crime labs, using different tests. Some items might have had 5 different tests run on them. So one test from the Promega company called PowerPlex 16, another company from Applied Biosystems called Profiler, maybe a mini filer kit that was developed to look at degraded DNA. That's why there were over 100,000 DNA tests done, even though there were only 18,000 victim remains. We have a lot of extra data that could be used to figure out this problem. So what's the problem with limited genotype inference, the old style approaches that are not probabilistic genotyping? Why is it that these older absolute methods cannot interpret imperfect data? Most DNA data is imperfect. There are many, many reasons. A lot of papers have been written about this. Studies have been done. I'll just give you a few that are relevant here. Well, their beginning is they start with the input and they take the data and they start throwing it out. They apply thresholds, they remove quantitative information. If you have a tall peak in the data that indicates a huge amount of DNA, that's considered to be the same as a moderate amount of DNA. The quantity isn't used. The process starts by throwing out the evidence, and that's not good in science, to throw out your data. The output is wrong. It's not genotypes. We're trying to compare genotypes. Instead, these primitive methods only look at the building blocks, the alleles, and that loses most of the information. Decisions are made. The analysts have to continually make choices about the data, parameters, what they do, how they operate, and those decisions can totally reverse the answer, making inclusionary results exclusionary and vice versa. The DNA data is changed and modified. The approach is black or white, all or none. It's data that fits a mental model of an analyst, or it's thrown out and the information is never used. The results are usually nothing or produce inaccurate match statistics. And the tests are done one at a time. There's no combination. If you have more data, it can't be combined. That's pretty limited. But modern genotype inference with probabilistic genotyping methods can interpret any data. Now, what if the data is totally uninformative? Then your match statistic is uninformative. But when the data has any pulse at all, and it's trying to tell you something, then as you see on the right column under probabilistic methods, now you're actually moving forward with this analysis. The input can use all of the data. Nothing is discarded. The output is in genotypes and their probabilities, considering all possibilities. There's no decisions to make because everything is considered, all the data and all the genotype possibilities. The data is preserved, not discarded. There isn't a concept of trying to choose what data somebody thinks is reliable. Rather, what explanations better explain the data we have without touching it. The approach is not black or white, it's shades of gray, it's probabilities. And the results are accurate answers, as confirmed in, at this point, probably in hundreds of validation studies across all the different methods that have been developed. And the tests are not just one at a time, but if different laboratories have analyzed the same sample with different tests, then all that data can be used. Let me tell you about that, because that was very important in looking at the victim remains when Cybergenetics analyzed them in our TrueAllele technology. Joint data analysis is standard in science. It's why pharmaceutical companies, when they test drugs, don't look at one rat and the FDA approves it. You look at many tests, many rats. You repeat your experiments. You look at different subjects. That's normal science. And to do that, you have to be able to statistically combine the different data experiments. Here, the different tests were in the form of different kits that manufacturers ran. Profiler Plus, Cofiler, PowerPlex 16. The Bodhi company had their own multiplex kits, Bodhiplex 1, Bodhiplex 2. There were mini filer kits where people were looking specifically for degraded DNA. But by having the computer able to use all of this data simultaneously, it was able to get more information and get stronger associations from those more definite genotypes. So this is an example of combining data from a study we had done around that time, over 10 years ago, where instead of looking at one swab from a handgun, the handgun was swabbed in 4 different areas, and they're shown here in red circles. And in fact, the lab tested each area twice. So instead of having one test, there were 8 tests. And what this published work showed is that as you kept adding more tests, you went from some information from one test to almost complete information when you combined all the tests. And we did the same thing with the World Trade Center data. We would identify which of the tests corresponded to one sample, to one victim remain item. There was mislabeling, so that actually took a lot of work. But once we found, say, the 4 or 5 DNA tests that were done on one item, we would have the computer run those all together to get much more information, a more definite genotype with less uncertainty in it than if we'd only done it one at a time. The project also involved looking at the World Trade Center missing people. We didn't have the people they were missing, so we couldn't compare with their DNA. But we did have their relatives, and we had personal effects. The relatives share inherited DNA, and there's rules of genetics and inheritance that let you write down the mathematics and statistics and solve for genotypes. Now, interestingly, originally, the first responders who were collecting the family's DNA didn't collect spouse DNA of a missing person. That's because they knew that the person wasn't genetically related to the missing person. They were their spouse. However, if you're going to reconstruct a missing person's genotype, you have to know how both parents impact what the children inherited. So you can't fully analyze the children's DNA without having the spouse. So the crime lab went back, I think over a few years perhaps, to find and get the cheek swabs and the genotypes of the spouses. The personal effects were often mixtures. They were low level. And so from all of this secondary DNA, from relatives, from personal effects, what our task was to reconstruct the genotypes of the missing people from the DNA we had available, to compare those genotypes with the victim remains genotypes to see if we could make associations. So with Sarah has a family. And in the family, we see that she has parents, she has a husband, she has 2 children shown here. And from her family and from over 1,000 other families that we analyzed, we were able to reconstruct the genotypes from the family members. And we studied this, and I think I presented this at a conference over 10 years ago, is that it's the same concept of data. The more relatives you have, the more information you have. If you only have one child, you'll get a certain amount of information. If you have 2 children, you'll get typically more information. And as you add more relatives, you keep getting more and more information. On the left, I'm showing a pedigree. It's not exact. S is for Sarah, the missing person. It's not exactly her pedigree because there's a 3rd child under here, but the row at the top are the parents, the black dot next to Sarah is her husband, and the squares underneath would be her children. From their genotypes and this pedigree, there's a flow of information of how DNA has been inherited from ancestors and how it's influenced your offspring. And mathematically, you can then reconstruct Sarah's genotype. The graph on the right, from the study we had done, looking at what happens when you have more and more relatives, we see that the first column with one relative produces a certain amount of information. But the scale here vertically is the number of zeros in the match statistic. 6 for a million, 12 for a trillion. I think it goes up to about 16, which is almost a billion. So it's the number of zeros, which is how scientists measure information. And you see as we go 1, 2, 3, 4, 6, putting in more and more relatives, in the genetic reconstruction, the computer gets more and more information until finally, with 6 relatives, we're almost at the right column, which is the full amount of genetic information. So these genetic reconstructions that our system does is very powerful in working with family members to reconstruct the missing person's genotype from the relative's genotypes. There are also genotypes from personal effects, toothbrushes, razors, clothing. And this is also useful information. They're not like cheek swabs or reference samples that are pristine DNA. The DNA here can be in lower quantity, it can be a little degraded, it can also be a mixture. People might share toothbrushes or clothing, and we had hundreds of mixtures that we needed to analyze in the World Trade Center project. So part of what I invented 20 years ago was how to unmix mixtures. It was done in a very uninformative way with human review, but with probabilistic genotyping, we could use all of the information, account for the PCR defect, and get accurate probabilities. And I'm not going to tell you about how that's done. You can read about it elsewhere, look at our website. But the concept of it is imagine that we have, say, 3 people's DNA all mixed in together in one item. I'm representing that here as a picture of 3 women. And they're all mixed in together, and what we want is the genotypes of each one, each of the 3 separated so we can make comparisons. That's what the computer does. It separates out their genotypes. I'm showing the pictures a little fuzzy or faded because it's up to probability. We don't know them exactly. But these probabilistic genotypes are very informative, and they've been routinely used in hundreds of thousands of cases now from the different probabilistic genotyping products that are out. It's basically why complex crime scenes, sexual assaults, just handguns and so on can even be analyzed today instead of the evidence not being used in criminal justice. But in this World Trade Center effort, we had to unmix mixtures. But that's not all. So yes, we can reconstruct genotypes from relatives and their DNA, and we can reconstruct genotypes from DNA mixtures and low-level or degraded DNA. But what we did for the World Trade Center is we combined both methods. So what we were doing was to say there's a certain amount of genetic information from the relatives, there's a certain amount of personal or phenotypic information that we can see from the personal effects or the mixtures. Let's use all of that information, the genotypic and the phenotypic, the relatives plus the personal effects, to get as much information as we possibly can from relatives and effects about the missing person. And here you see we're reconstructing Sarah using everything we have, whatever she's owned, the people she's related to. And this reconstruction, again, it's not the exact picture, but I'm showing little icons. There's her family in the bottom left. They contributed data. There's some personal effects like a toothbrush on the bottom right. We've reconstructed her genotype. So we're looking at, this is a picture of her genotype from this type of information, reconstructing Sarah. Well, once we've reconstructed and inferred these uncertain genotypes, we have inferred victim remains genotypes, shown on the left with the victim remain in the picture, and we have an inferred missing persons genotype. Here we see what we've reconstructed with Sarah from her family and her personal effects. We can now make a comparison of these uncertain genotypes to provide a definite, certain match strength. We can get a match statistic that tells us how strong the association is. And that statistic, all the math is saying is that it's using all the genotypes now. We don't have a reference, so it has to consider the strength of association relative to random from the victim remain genotype and the missing person genotype relative to what coincidence would be, all genotypes need to be considered, but the math doesn't care. It'll be hard for a person to do, but you can imagine a computer can do a calculation that's basically multiplication, division, and addition. And they do it very quickly. So here we are associating from the left the inferred genotype from Sarah's victim remains. We don't know it's Sarah yet, that's the goal. And on the right is the inferred genotype of Sarah from her relatives and her personal effects. And notice here that there is no reference sample. This was fairly revolutionary for 20 years ago, where forensic science was based on match statistics using reference samples. There's no reference samples. We're just comparing evidence to produce match statistics to tell us, is there a strong association, a weak association, no association, and so on. So let me tell you about the True Allele system. We had the True Allele method, and we were starting to build different systems. But when the crime lab reached out to us from New York and said they would like us to reanalyze all of this data, we needed to build a supercomputer. So we did. And the supercomputer was built around a database. We had been playing around with using computer files for 5 years, and it was pretty clear that you could not use computer files. There was just too much data. You needed a database that could house, organize, and store all the data and the results and do a lot more. So we began by building a 24-processor supercomputer over in Oakland here in Pittsburgh in our office, and we built a matching database. I'll tell you more about what that does. That's shown on the top in blue. We took a bunch of off-the-shelf processors, put them together with a database, and had them do the matching and hold all the data for us. So it's a matching database. On the bottom right, we also had a stack of computers that could do the genotype inference. If we gave it data from victim remains, from relatives, from personal effects, it could crank out these genotypes up to probability and put them on the database for us. And we also had, shown on the bottom left, was user interfaces. Again, multiple interfaces. We had a number of Duquesne interns working on the project at the time. And they were needed to get the data into the system, look at it briefly to make sure that it made sense. There were tasks of making sure that the data was from the same item to overcome clerical errors. We had protocols which reached the point where to overcome minor errors that had been made, because the whole process really was automated, but we needed to do checking to see if we're looking at the right samples. Students would take about 40 seconds per victim remain to get the data into the system. So for entering the DNA data, the analyst would be trained. They would upload the data that was related to a sample or victim reference, like personal effects for a missing person. They would also upload questions, which were called requests. They were automatically constructed, but the question was, here's the data, what question do you have? And the question is, generate the genotype for us, find out what that is. And they would do a quick visual check to make sure that they weren't combining data from different victim remains. Again, we had multiple tests. We could have co-filer and profiler and PowerPlex 16. So if we're going to have the multiple tests on a sample all in together and make the best use of the data we had, we did want to do that joint analysis, but we didn't want to do it incorrectly. So people would enter the data and ask the questions. The matching DNA database did more than just setting up the match. It also did a lot of coordination. It was set up to be a database, but also a database that ran and monitored all of the genotyping computers. If anything went down, it would fix it, bring it back up, it would handle updates. There was a little expert system that we built inside the database computer to monitor all the other computers as well as do the matching. So it would store the data and the questions, it would store the genotypes that came back, and then it would do the comparison. For inferring genotypes, on the bottom right, we have our genotype inference being done by this parallel computing of 24 core processors. And by having parallelization, we can go a lot faster. In our office now, we can run 100 processors at the same time, which means we can solve 100 problems in the space of the time it takes to do, would have taken to do one. And this probabilistic genotyping preserves the identification information. It's not making choices, it's using all the data. So moving from those parallel computers, when one of those processes are done answering a question, that arrow moving to the left shows that we now have a genotype, say from a victim remain that corresponds to Sarah, or for a missing person, like Sarah's genotype. These are fuzzy probabilistic genotypes, but they can be compared and get strong match numbers when we're done. And finally, the matching database can actually match these genotypes. It compares all of the victim remains genotypes shown on the left, they're Sarah's, with all of the missing persons genotypes from family and personal effects, there's Sarah's shown on the bottom right. And it's a very powerful matching algorithm. It's automated. There's really no person involved. The genotypes come in, and the system is triggered by the arrival of the genotypes to do the work. There's no person. It's also incremental. It's smart enough to know to only match the new genotypes against old and new genotypes. It doesn't redundantly do any work. It's actually based on an algorithm that was developed at Carnegie Mellon in the 1980s called the Reedy match algorithm. And it's just incremental matching. It's very powerful, saves a lot of time. And it's highly informative because the matching is not like the very weak allele matching that we have in government databases today. Instead, it's this highly informative genotype to genotype matching. Again, I write this LR equals this math formula. The same way that the probabilistic genotypes are preserving all the information from all the data that they analyze, from victim remains or from missing people, the number that's being produced as the match association is again using all of that information from the data through the genotype. So the data is telling us how strong the match is. And the database just keeps matching and making comparisons continuously. So the result of doing all of these comparisons, of comparing the 18,000 victim remains genotypes with the 2,700 missing persons genotypes, is that we can identify the victim remains. And so there, on the left, there's an identified victim remain. In this story, it corresponds to Sarah, and then Sarah's victim remains can be returned to her family for closure. And from the family's perspective, that's partly the point of why all this DNA work has been done. From the medical examiner's perspective, they can now close the case, person's been identified, and their death can be registered. We worked with the lab to validate TrueAllele. When science, reliability is based on testing, so you test methods to determine whether or not they work or how well they work. We conducted a concordance on 150 comparisons between World Trade Center victim remains and missing people, and Cybergenetics used its TrueAllele method. New York City used its methods. These were simpler data because the more exotic data the lab didn't have methods for. The lab presented its research findings in a talk, the American Academy of Forensic Sciences meeting in Georgia in 2012. And then with concordance established, Cybergenetics was then able to file all of our World Trade Center comparison results, where we would list an identifier for the victim remain, an identifier for the missing person, and what the strength of match was, and provide that for these roughly 50 million comparisons, what was the subset that actually produced an association. So this TrueAllele database that we built for the World Trade Center is quite a scalable database, and it's evolved over the last 15 years. It started off as a computer rack-based system. We were able to take everything that was on the rack and reconfigure it and squeeze it into a pretty powerful parallel desktop computer, but it can fit on a desktop. That's what crime labs tend to use when they run TrueAllele. And in the last 5 years or so, we've moved the architecture up to cloud computing, so we can run it up in the cloud. We just can spin up these databases and servers and interpret machines as needed. And that means we can scale to any size problem. If another problem that involved making 50 million comparisons came up, we would have our computers up and running in a matter of hours to be able to have enough computing power to examine all that data. The system, TrueAllele, has evolved. We run on all modern DNA markers for shorthand and repeats, and other markers as well, and we've now incorporated next-generation sequencing data, NGS, into the system. The platform can handle any type of genetic locus data. The system's designed to be completely automated. We do have people involved for casework, because in the end someone does have to testify. But the system was designed to work without people. The data can flow in, questions for solution can be generated automatically, and the coordinating database ships them out to the 100 processors that are waiting to solve, takes the results back from the inference computers, makes comparisons, and records and can report the match statistics. It's basically, in the end, the final step is a genotype matching database. All TrueAllele can do is infer genotypes and match genotypes. It just does it in a really powerful way. It infers genotypes in a way that preserves all of the identification information that was in the forensic DNA data, and it matches the genotypes as if it were the data, again, preserving all the information. And it's designed for and around, from the outset 20 years ago, for probabilistic genotypes. It's not an add-on. Obviously, definite genotypes are also probabilistic. They just have a probability of 1. So it's designed for anything that's a genotype. TrueAllele's been used extensively in criminal justice. It's used by prosecutors, by defenders. We've helped exonerate 10 innocent men with the system. Reports have been issued by Cybergenetics in 45 states. Obviously, our crime lab users issue their own reports. There have been over 40 validation studies demonstrating its efficacy, 8 of them published in peer-reviewed journals, which is a sign that the community accepts the results. There have been over 30 admissibility decisions where, say, a defendant challenges the scientific reliability, and then a prosecutor presents evidence and witnesses to show why this is a tested, reliable system. TrueAllele has been used in tens of thousands of cases, and if you look at all the probabilistic systems out there for genotyping, it's been used in hundreds of thousands of cases now. And in many of these cases, there would have been no DNA evidence otherwise. These 10 men would have stayed in prison. It's just better science can find better truth and better information that ideally leads to better justice. I do want to tell you about a project that was done in Cleveland by one of our labs that uses TrueAllele. They reported on it at the American Academy meeting using their TrueAllele database. They really like their database. They use it quite a bit. This was a project where there were about 150 auto thefts by juveniles. And it was the type of data that really isn't used by crime labs because it's evidence data, touch DNA, mixtures, low level, uninterpretable by most manual review protocols. And it's not that you even have a person to compare it with. The question was, could all these crime scenes be associated if the same person left their DNA? So these are evidence-to-evidence comparisons, and the computer dissected the entire crime spree, connected the different stolen vehicles, and in the end was able to connect it to known perpetrators. So this is something that can be done using an automated information preserving database. So TrueAllele is now set up for any sort of databasing, whether it's familial searching or property crime or solving cold cases, but it's also set up for mass disasters. The matching database shown on top is now a parallel engine. So if we run it with 100 processors, it runs about 100 times faster. You can just do that much more, higher throughput. The user interface on the bottom left can do a lot more. For example, reporting error rates, which are useful in science and usually not reported by crime labs, but are very helpful when you're reporting low match statistics to give a context of how often would somebody who's not involved in the evidence be seen. That really helps juries understand evidence. And then on the bottom right, we have the genotype inference. The inference has gotten more and more powerful and more validated. The most recent validation paper shows that TrueAllele can reliably separate mixtures containing up to 10 unknown individuals. That's a lot of people in one small amount of DNA. Basically, if you have anything at all, or if you have evidence that exceeds what a crime lab is capable or willing to do, groups, including crime labs, will send the evidence to Cybergenetics for consultation and TrueAllele processing. But this is set up that in the event of, God forbid, another major mass disaster, as the data starts getting generated, the connections between the victim remains and the missing people can be automatically produced as computer information. If you'd like more information about the system, the approach, the methods, there's hundreds of pages of talks, videos, papers, conferences, presentations, webinars, and so on our website and our YouTube channel. And I think I'll stop there and take questions. Thank you very much.